In human development, all development starts from the moment of conception. And you need to know the process of human conception. Now, you are all familiar with the male sperm, which is the sex cell coming from the father, and the female ovum, which is the sex cell coming from the mother. And during conception, you can read all the detail, which is interesting, but during conception, the male sperm penetrates the ovum of the woman, and in that way fertilizes the egg, and the two nuclei, the core of those two cells, then unite to form one core in a new single cell which will start multiplying. So that is the process of conception. And as you see there, in the fallopian tube of the woman, the sperm comes from the male, plus the ovum from the female leads to conception. The second sentence that you see there is a sex cell is called a gamete. Gamete. So, you have a sperm gamete plus an ovum gamete, and they unite and they form new life, which is a zygote. But that is the basic process that you need to understand. Good. After that, new life starts building, starts forming, starts creating. It is the building blocks of such a new life. A new life is built by the 23 chromosomes coming from the father, or the sperm, and the 23 chromosomes coming from the mother, or the ovum, and they unite, and then the zygote or zygote has a combination of 46 chromosomes. The human zygote or zygote has 23 pairs of chromosomes, and you now know how they were combined. Secondly, there you see the sperm contributes 23 and the ovum 23, and that makes up the 23 pairs or the 46 chromosomes. The second building block would be the genes. Okay? Genes make up chromosomes. Let me see if I can explain that to you. That is, in fact, not a very nice one, but more or less, that is the double helix that we call a chromosome. And genes you will find within these spaces that make up the double helix. Okay? So, in fact, genes build chromosomes. Okay? And the genes are the smallest units of heredity, and they are the carriers of all inherited characteristics that will in the end build that particular individual or person. Genes come in pairs called alleles. So genes, very important there, and the genes will really be the carriers of what will make the human person. Good. Then we go to the chemical substance, the chemistry that really activates all the interactions coming from the chromosomes through the genes into the cells to create the person. And the DNA is a chemical substance pronounced deoxyribonucleic acid. Deoxyribonucleic acid, or DNA. As I said, the DNA is the chemistry of chromosomes and genes. It is the blueprint for all human development. It is the recipe or blueprint for all cellular function. Okay? And all of us, all human beings on earth, doesn't matter what your creed or race or skin color or whatever is, we share 99.9% of our DNA. So we are much, much more similar than the few differences that we may have. Okay, that's as far as DNA is concerned, being the chemical that gives all the developmental instructions. But DNA also works with another acid called the RNA, which is ribonucleic acid. And it's a protein made by the DNA to perform the functions. It carries the blueprint information from the cell nucleus to the cell body and cytoplasma, where the work really starts. Okay, that in a nutshell is what you need to know. Those are the building blocks. Next, for this study unit, after the building blocks, we go to cell division and crossover. Okay, normal cell division is called mitosis. That happens in every single normal cell in your body 1,000,000 zillion times throughout your lifetime. Okay? So, what happens there? The chromosomes are in their 23 pairs. Okay? Those are the normal pairs of the chromosomes. In mitosis, you have a division of the chromosomes roughly through lengthwise, through the half. And then the one part of the chromosomes will move to one pole of the cell, and the other part will move to the other pole of the cell. Then the cell will divide, and then you will have the two cells with all the information that the previous cell had. A student once said to me, now, if the chromosomes are cut off here, what happens to that? What simply happens is that the DNA and RNA within the cells simply heal the chromosomes because they have enough genetic information to be able to do that. So in every single cell, the chromosome simply extends again and heals, and in that way, new cells are created, once again carrying 46 chromosomes with all the necessary information. Okay, that would then be mitosis, the normal division of cells. We say there, normal cell division. Division of all chromosomes lengthwise to make 92 chromosomes, but once the cells close and split, they will be back to 46 chromosomes. The halved chromosomes move to opposite poles, and the cell divides, creating two new cells with all the information. Okay, that is normal cell division. In meiosis, you simply have, once again the chromosomes, and now they are in their pairs, you can see there. Now the chromosomal pair then breaks up or moves away, so that, and the one moves to the pole, and the other one moves to the pole, and then you have a new cell formation once those cells divide. But in the new cell, for meiosis, you will have each part would then have 23 chromosomes, because those are the sex cells. They must then be contributed to the next generation as a contribution either from the father or the mother to make up the 46 chromosomes once again. Let's quickly recap. Mitosis, normal cell division. Meiosis, sex-cell division. Mitosis, 46 chromosomes in each cell. Meiosis, 23 chromosomes in each cell. Okay? Then we quickly have a look at the crossover. Crossover simply says that chromosomes are very creative and they're very eager to share their information. So, if you have a pair of chromosomes, for example here, then this part of the pair is eager to share some of its genetic information, namely the genes, with this part of the chromosome. And simply, in dividing, they then carry over of their information to one another, and that enriches the new cell and carries over interesting genetic information. So that means that you are always more than just your mom and dad. Okay? So your genetic inheritance is always much more, much richer than just what you have received from your father and your mother. Okay? In fact, your genetic inheritance has something like 27.8 million possibilities in which you have been created. And that makes, last week we touched on the uniqueness of every person. That makes also you and I so unique that it's not simply a duplication of mom and dad. It is much, much more. Okay, that is crossover. Good. Then we quickly have a look at dominant and recessive gene pairs, because this is now the way in which the information will be carried over and characteristics will develop. Good. So there we see that genes come in pairs. We have already said that. And for every pair of genes, you could have two very similar or identical genes, which would then be homozygous or homozygous. Okay? And if they differ, it would be heterozygous. Okay? If you're happy to have two homozygous genes in a pair, then the characteristic is very clear. You will receive the characteristic carried by both those genes in the pair. It becomes a bit more complex when you have one or two heterozygous genes in a pair. Then there's a little bit more confusion. But then the very important principle kicks in that we know to be the principle of genetic domination or recessivity. Okay, so many of our cells, or all our cells, are either dominant cells, so they have muscles. They are fighters, they're dominant. Or we have recessive cells, and they're the more meek and mild ones. Okay, so all our main characteristics will come from dominant genes. So the dominant genes will always dominate and carry over the characteristics that they represent. Okay, so even if a mom and dad or a man and woman, if the man has a dominant gene and the woman a recessive gene, the dominant gene will carry over and create the characteristic. Okay, good. So, if you have two dominant genes, then it will be a double D, two dominant characteristics. If the man has a dominant gene and the woman has a recessive gene, then you have a D and a small d, but the characteristic will still be carried by the dominant gene. Okay, so when will the recessive genes do something? And that is only if two recessive genes combine, then they will carry the characteristic. Okay, good. That is as far as dominant and recessive information carried over. Then we quickly have a look at the end of mind map 1, namely sex determination. And I can clearly see that many of you have done biology, so this is no surprise to you. But for many years in these classes, when we discuss this, students do not want to believe me if I say, if a couple wants a boy or a girl and the other sex is born, who is to blame? The father. Yeah. Not the mother. Not the women. And unfortunately, even in this modern time, there are still many, many cultures where women are blamed when they produce a child which is different from the gender that had been wished by the family or by the father. And in certain cultures, such women may even be killed if they produce and that is so unfair, because I hope you clearly now understand that it is the male, it is the father that determines the gender of the child. Okay. The male chromosome is an XY chromosome. The female chromosome is an XX chromosome. So remember, a child can only receive an X from the mother. That is all that she has to give, because her chromosome has only X's. Are you with me? The father has the Y. So if one of his gametes or his sex cells meet with the ovum of the woman, and in his sex cell there is another X, because that's also part of his chromosome, then the two X's unite and it will be a girl determined by the father's contribution. If, on the other hand, the father contributes in his other sex cells a Y chromosome, because you remember the chromosome split. So in his cells would be X's or Y's, and they have equal chances. So if he then contributes a Y, the Y part of his chromosome meets with a woman, unites with the X of the woman, then you have a XY, which is a boy. Okay? So very clear now. Just remember that gender is determined by the sex cells of the father. Okay? Who is to blame? Yeah. And in the second mind map, we quickly have a look at what we understand under multiple births. So what happens when a woman has more than one child? And there are two possibilities. The first is, you can have a look. There are 5 factors that could and would determine whether a woman has multiple children in one birth session. Namely, whether she had infertility treatment, that raises the possibility of multiple births. You also see there the family history, the the maternal age, older women more often. You also see the race in certain races more often, and then largely the maternal size. In other words, the body size of the mother can also contribute. So those are the factors influencing the possibility of multiple births. Then, how does it happen? The first opportunity, or the first possibility, is where you have a monozygotic twin, meaning that one sperm met with one ovum and fertilized one ovum, and then the ovum divided into two cells, and two identical persons were formed. Okay? That is monozygotic twins. And they are always identical. They are usually of the same sex, and they carry identical genetic information. Good. What happens then if you have fraternal twins that are not identical? And there, two ova in the woman were fertilized by two separate sperms of the male, and two separate zygotes developed. Okay? And their genetic information would be not as similar as identical twins. In fact, they would be nothing less than normal brothers and sisters born at the same time. Okay? What happens when we have conjoined twins, or what sometimes happen as the so-called Siamese twins, where the babies, a twin, usually identical, are born, but they are joined at limbs. Okay? They should have been an identical pair of twins, because there also it is one ovum fertilized by one sperm. The cell divides, but it was unsuccessful cell division. And therefore, they are then joined and often develop shared organs. Often they have shared parts of hearts, parts of their heads, even lungs and digestive systems. Okay? So they are similar than monozygotic twins, but it was a unsuccessful cell division, and therefore they remained conjoined. That is multiple births. Let's have a look at genetic abnormalities. Yes? Although our genes are very powerful, very strong, very creative, things can go wrong. So, we can have abnormalities in people as a result of, in the first place, gene abnormalities, and secondly, as a result of chromosomal abnormalities. Okay, in gene abnormalities, it simply happens that the genes convey faulty information. Something goes wrong with their DNA and with the functions of the RNA, and they cannot convey the correct information. And in that way, you can have genetic or gene abnormalities. In gene abnormalities, we could have abnormalities that are simply only carried by women, some only by men, and therefore it would be called the sex-linked gene abnormalities. You could also have certain gene abnormalities that happen within ethnic groups, and then it would be contributed by the dominant genes running in a particular ethnic group, and that would be your gene abnormalities. The next one would be chromosomal abnormalities, and a very common one here is Down syndrome. where on the 21st pair of chromosomes, you have not only the 2 chromosomes, you have an extra chromosome. So, Down syndrome people have 47 chromosomes, with 3 on the 21st chromosome pair. And therefore, they are usually mentally retarded, they have very specific features, etc. Okay, good. Another example of a chromosomal abnormality could be where you have 3 X's in the chromosome. And those are also usually highly mentally retarded, very difficult people. They usually die at about 2 to 3 years of age, but that would also be a chromosomal abnormality. You could also have an XXY with 2 X's and a Y. That is called the so-called Hunter's chromosomal deviation, and they also would be very difficult to live and would most likely not live long. Then lastly, just an example, you could have a chromosomal pair or chromosomal bonding of an X only, and it's usually written X0. That means the other X is missing or the Y is missing. Okay, and they also die very young. Those are just examples of the so-called chromosomal abnormalities. That then is as far as the main genetic building blocks in our development is concerned. Now we can quickly go back and have a look at the physical development after conception of the baby. Good. And that's where we then go to the prenatal stages of development. By now you know that prenatal is before birth, and it runs from the moment of conception to the moment of birth, usually 9 months, or varying between 266 to 280 days. Okay? That is the so-called prenatal period. In the prenatal period, there are 3 developmental phases or stages. And the first one is, as we have just now said, the germinal stage, in which the zygote or zygote is the product. And it's from conception to the 2nd week after conception. Okay? And in that phase, what is most important is that the zygote begins to form, or the zygote. Mitosis begins about 24 hours after conception, as you can see there, up to 36 hours after conception, the real cell division begins, and then it increases at an enormous pace. Within, under the germinal stage further, the zygote, the cell, starts moving from the fallopian tube into the uterus of the mother and attaches itself to the inner wall of the uterus, where it will grow, where it will be fed, etc. And that attaching process is called implantation. It implants itself. Okay, so the germinal phase is from conception until the zygote had attached itself to the uterus wall. Good. Then we go to the next phase called the embryonic phase or stage. It is usually from about from the implantation up till 8 weeks after conception. So from implantation up to 8 weeks after conception, and 8 weeks is usually the time that the woman can find out that she is pregnant. Okay? So usually by the end of the embryonic stage, the woman realizes and gets the information that she is pregnant. Let's quickly have a look at what happens in the embryonic stage. The embryo develops along the line of 3 layers, and that's very important. The first layer is what we can compare with a shell. In other words, it's an outside mass to keep the insides in. So first of all, you have a development of the outside shell, also called the outer layer or the ectodermic layer. And usually in that layer, as you can see there, are very specific outer layers such as skin, etc., taking place there. But it has the purpose of developing an outer layer which can contain all the rest of the development which is to come. Good. That's the first one. The second one would then be a sort of a middle layer, the mesodermic layer. And in the mesoderm develops the structure. So, as the container, the shell had developed to keep the insides in, after that, a structure will develop that can sustain the development. And that structure usually then contains the basic bone structure, the basic skeletal structure, and some of the muscles to keep it together. So can you see? First one is to keep it inside. The second one is to give a form, the structure. And the third development there would be your endoderm, the outside layer, the inside layer, and that would usually then be all the life-sustaining organs, part of the heart, the respiratory system, the lungs, the liver, the digestive system, and things like that. Okay? So, in the embryo, there are 3 very important stages: development of the outside shell, development of the middle structure, and development of the inside parts, which would be the life-sustaining organs. Good. In the embryonic stage, you also need to understand that 2 very important organs form. And that is, in the first place, the umbilical cord, which is the cord linking the embryo to the uterus wall of the mother to be fed and nourished and receive oxygen. And the placenta, which is the lining of the uterus wall of the mother, and that is the organ that provides the nourishment and the oxygen to this developing embryo. Okay. In a nutshell, that then the embryonic phase. Then we come to the fetal stage, and that is usually from 8 weeks to birth, and normal differentiation of cells and development of organs take place in the fetal stage, during which the 7th month is very important, because from 7 months the child, the baby, is viable, can live, okay, can survive if there is a premature birth. So a fairly important time of the development of the fetus would be at 7 months. Okay? That is about the most important that you need to know. And after 7 months, the development is really in preparation for birth. Lastly, for this second chapter and second study unit, you have all the things that can go wrong, that can create enormous complications for the development of the unborn child. And there you see what can go wrong. For example, in the first place, the age of the parents. The very young mother, in other words, puberty or very young adolescent mothers very often have serious problems in their pregnancies, simply because their reproductive organs are still fairly young and fairly undeveloped. The same, the opposite would be for the older mother. Usually 35 years for a first pregnancy and about 40 years for any other pregnancy, that would start being a risk due to also the same reasons the reproductive organs of the mother had aged and had lost their elasticity and their feeding power. And that could create serious problems for the unborn child. Okay? So, and then it's now fairly new information says that the age of the father, we're now beginning to know that the semen or the sperm of males anywhere from 45 to 50 years of age are losing their quality. So also the age of the father has recently become a risk factor. Good. Then secondly, we see the nourishment of the mother, nutrition of the mother. You can read that. As I said, the infant needs to be fed from the bloodstream of the mother through the placenta. If the mother is undernourished, the infant will certainly be extra undernourished. So the nutrition of the mother is extremely important. And malnutrition has many, many negative or adverse effects. But recently we also realized that malnutrition could lead to slow brain development, to a lower brain development and cognitive thinking skills, to lower intelligence, and for a lower brain mass or weight, and many such very serious problems. So the nutrition of the mother, very important. Okay? Next, we quickly then have a look at the medication, or sorry, radiation first. Radiation, and you know, pregnant women should not have x-rays taken because the radiation can really interrupt very seriously with cell division, and radiation can also destroy the DNA. So up to at least about 3 months, radiation can be very adverse for the growing baby. Then you have diseases of the mother. The unborn child is mostly protected from all viruses and bacteria through the placenta. The placenta is very protective and very powerful. However, certain viruses can penetrate the placenta and cause severe damage. And by now you all know that the HIV virus is so powerful and strong that it can penetrate the placenta and cause severe difficulties for the developing infant. However, most infections of infants to HIV-positive women take place during the birth process itself, where there are moments when the blood of the mother and the blood of the baby sort of exchange, and most of the infection then takes place there. Okay? You can read about that, and we know that there is a very scaring statistic of that in South Africa about 6,000 HIV-positive babies are born every month. So it's very concerning. You will also read that the mother's milk, breast milk can also infect the child. Good? So that is as far as HIV and AIDS is concerned. The next one is rubella, the so-called virus that causes German measles. Very, very, very dangerous within the first 3 months of pregnancy, will cause blindness, deafness, and mental retardation and slow development in the baby, the unborn child. Okay? Rubella is the next one. Then we have a look at also a sexually transmitted disease, syphilis. Syphilis, very, very serious infection, usually from about 4 to 5 months. For the first 3 months, the placenta is strong enough to protect the child, but usually from 4 to 5 months, the syphilis organism, it's not a virus, it's an organism, can penetrate and then create enormous and serious damage to the unborn child. Okay, you can read about that. And then lastly, genital herpes. And this infection usually takes place during birth, when the child is in the birth canal of the mother and comes into contact with the herpes virus. Okay? Okay. Then we quickly have a look at the medication and drugs used by the mother. Medication and drugs mostly cause slow or low fetal growth. So it interferes with the basic growth and cell division of the child. And the most important there would be the fetal tobacco syndrome of smoking mothers and the fetal alcohol syndrome of drinking mothers. And those effects would be highly, highly adverse and negative. Drugs that the mother take, similar effects. Also, they cause very often brain damage, very often mental retardation, the slower growth of internal organs. And what is very tragic, and it's usually very sad to see, is when a baby is born of a mother addicted to drugs, the baby will have withdrawal symptoms so serious that they will have difficulty breathing, or they will have brain convulsions and often die immediately after birth, due to the withdrawal symptoms, which are very, very strong. Okay. And then lastly, we have a quick look at the emotional state of the mother. When a mother is in a negative emotional state, she secretes hormones into her bloodstream. The hormones go through the placenta and into the baby, and it immediately influences the movements and the activity of the baby. And what is sad is, after a few hours or a few days, the mom perhaps have calmed down, and she's okay, whereas the infant, the baby, is still activated by the drug, by the hormones of the mother. So the negative part of the influence of the mother's emotions on the child is that it has a prolonged effect on the child, even after the mother has calmed down or feels better. Okay? And now we know that stress, but especially high levels of anxiety in mothers, cause very serious negative effects for the babies, for the infants. Okay. See you next week.